Thanks again, Becky.
Welcome, everybody. Thanks for joining us today for Family Office Insights Mercury series, where we partnered with Mercury, the neobank in San Francisco, to highlight some of the more successful firms that happen to be using the platform to do their banking activities. And as many of you know, we've had several of these, and we look forward to having several more, and delighted to host them in partnership with Mercury for the Family Office Insights Luminary Series. We are going to be recording this, and so if you missed something or you want to visit it later, you're welcome to do so. And of course, as always, if there's people who you know, your colleagues, your network that you think would find it interesting, you're most welcome to share that with them, as well as you're at liberty to connect with Adam and his group directly if this is something you're interested in deploying capital in. And so with that, thank you all again for being here. We can't hear or see you. We know you're there. And I would recommend that if you have a question, please post it as it comes to mind. And we'll get to as many as possible, as we have a bunch of folks coming on today. And if we don't get to it, we'll be able to get to it post-webinar. So again, thank you for joining us and welcome. We're going to start out with Mike Malenfant from Mercury. I just want to mention that he's not the second string, but Shelley is generally here with us. And so she is traveling. And so Mike has kindly stepped in to talk about our partnership and Mercury's contribution to some of these extraordinary companies. Thank you, Mike. You're on mute.
No, appreciate it. I'll happily take second string. No worries there. I'll keep it brief. I recognize you all aren't here to talk with me today, but at a high level, I've been with the Mercury team for about 3 years now. I currently head up the early stage relationship management team, working with a lot of customers earlier in their startup journey or whatever that their experience might look like, focused on helping them as it relates to getting to that next stage or next step in their journey, and really focused on what Mercury can offer and supporting them as they scale to raise their Series A, Series B, and beyond. I haven't worked as closely with Arthur, as he alluded to, as my colleague Shelley, but really think of this partnership here as a way to add another angle or layer of value to our customers, and really be able to get them connected directly to, in this instance, obviously, Family Office Insights and the partnership that can be grown, I guess, from the seed that these conversations and this educational session plants. I'm super excited to learn a little bit more. I've actually got to work with Adam a little bit over the past year myself, and so I'm excited to hear the conversation. But without further ado, I think I'll kick it over to Becky, or Arthur. Did you want to chime in further from there?
Only in a disclosure way, which I neglected to mention, that Family Office Insights is a excited and delighted user of Mercury. So I just wanted to say that out loud, just as a disclosure and sort of endorsement. So we love working with them as a customer. So, sort of eating our own cooking here, so to speak. So Becky, take it away. Thank you.
Yeah, well, thank you so much for having me here. It's really a pleasure. I'm a partner on the A16Z Bio and Health investing team, and one of the lead investors for Arda's seed financing. By way of background, I've been with the A16Z team for over 3 years, and I'm a scientist by training. I spent time in both R&D as well as BD roles across pharma companies such as Genentech and Amgen before coming to A16Z. And on the team here, I focus on early stage biotech investments. I do need to give my own disclosure, which is saying that all comments I have today are my own and not intended to be investment advice. That being said, my partner Vinita Argawala and I were very enthusiastic to lead Arda's first financing about 2.5 years ago. And this was really for several reasons. So first, we found the overall thesis of Arda to be simple and yet big if true. So what if, like in oncology, elimination of entire pathogenic cells could lead to more deep, durable responses in cellular-driven diseases such as fibrosis? We loved the idea of leveraging years of modality engineering that had already been done for oncology and repurposing all of this work for large patient populations with high unmet need outside of oncology. And second, in these seed financings, we always ask, is the idea big if true? And are the founders the right people to do it? And so second, we could think of no founder who we thought was more qualified and more motivated to execute on this big idea. We found Adam to have a unique combination of disease area biology knowledge, deep relevant drug development expertise, very strong connections across both biotech and academic circles, and quite honestly, a true entrepreneur spirit. Adam really had a fire in his belly to execute on this vision. It's been an absolutely distinct pleasure to get to work with Adam over the past 2.5 years and to get to watch him execute on everything he set out to do and more. So we at A16Z are really thrilled to get to double down and to lead Arda's Series A financing. We can't wait to see what the future holds for not only this super special team, but importantly for all the patients that they're developing therapies for.
Perfect. That's really helpful. Thank you for sharing that with us. So my pedestrian view before you jump in, Adam, and I will admit that it's not at all learned in any stretch in biology, but it seems to me that it's pretty clear that what Adam is doing is beneficial, as it's attacking the bad stuff without hurting the good stuff. I mean, I know that's a very pedestrian view of what you're doing, but that sounds like what you're up to. So, Adam, besides telling me that I'm out of my mind and that doesn't make sense, could you tell us a little bit about what you're up to? And Becky, if you need to jump, go ahead.
You have to jump. Thank you so much for having me, and look forward to working with y'all.
Great. Thanks, Becky.
Yeah, Arthur. I mean, you said it right. And Becky also, I think, articulated the concept really well, which is we're trying to get rid of the bad cells and spare the good ones and modify disease biology. It's a simple idea. And when it comes to therapeutics, we think that the best ideas are often simple as long as we can then execute on them. And so you'll see as I present the data and the idea in this deck today that we really spent a lot of time thinking about what are the white hot risks around this approach? How could it go wrong? How can we buy those down as quickly as possible? And although, of course, there's still a lot to do before we get to a clinically approved drug, we've come a long way in the 2.5 years since we started the company. And thank you for having us here today. Thanks to everyone who's attending the webinar. I look forward to telling you more about the company.
Excellent. Well, let's get into it. Let's just talk a little bit about your background. Becky shared with us her enthusiasm with your skill and knowledge and so forth. I read about it, but could you give a Reader's Digest version for the group?
Absolutely. So I'm the founder and CEO of Arda Therapeutics. I've been operating this company for about 2.5 years. Before that, I spent about 7 years at a company called Calico Life Sciences, which Google started in order to study aging and age-related disease. And there I was a group leader and I led the development of a couple different therapeutics in the biologic space, one of which is now in the clinic. And really, before that, I was studying aging and age-related disease and cellular senescence in my PhD and postdoc at Stanford and Berkeley. And so this idea of thinking about how we can use cell depletion to target chronic diseases that aren't just cancer has really been in my mind since graduate school, when I was studying one type of pathogenic cell, which is a senescent cell. And it's been something that I've been excited to do for a long time, but didn't have the technological enablement to do it until very recently. And I'll explain why that is in the deck. But essentially, we have to be able to find targets that are specific. And we couldn't do that until we had a certain kind of data, single cell data, in sufficient quantity and quality to do this the right way. And in 2021, when I started Arda, it was because really we felt like we had finally reached that inflection point. And so that's my background. And well, I'll also mention just at the beginning, we're raising our Series A right now. As Becky mentioned, Andreessen Horowitz is leading it. We expect participation from a number of other insiders, including GV. And the ask is about $40 million total raised on a $40 million pre, so about $80 million post money. We have 25 million of that 40 committed, and so we're looking to raise the additional 15.
Excellent. Perfect. We'll revisit that later on to remind everybody, but thank you for doing that up front. So let's get into the meat of the matter.
Fantastic. I'll share my screen.
Yeah, let's see if that works here.
Let's see.
Oh, perfect. Perfect. Okay.
Okay. And please feel free to interrupt with questions. Should I monitor the chat or will you do that?
I'll take care of all of it. Yep.
Wonderful. Thank you. Okay. So the reason we exist is because drugs are getting worse and drug discovery is getting harder. And that's because for decades, we have employed a common strategy outside of oncology. In oncology, we kill cells all the time. That's what the majority of therapeutics do. Outside of oncology, we try to restore tissue health by blocking the activity of individual proteins and individual signaling pathways. And this can work. There are successful drugs that employ this. However, because we've been doing this for so long, and because modulating an entire tissue's biology with a single protein is a big ask, this is getting harder and harder to do effectively. Drug approval rates have declined dramatically since the early 2000s, and often the drugs that do get approved are only incrementally more effective than previous therapies. For a lot of chronic diseases that still have high morbidity rates and where patients really need relief, we think it's time for a more powerful approach. We want to target cells instead of targeting pathways. So that's what we're doing at Arda. We're pioneering targeted cell depletion. And we think of this as being a beneficial approach over traditional approaches in a number of ways. The first, I already mentioned, greater efficacy. We think this is just going to work better because we're getting rid of entire cell populations rather than individual factors within those cells. The second thing is there's an untapped opportunity space here. Nobody has explored targeted cell depletion outside of oncology. The one example that I can think of is B-cell depletion using targets like CD19 or CD20, but that's really just one example, and it's not even targeted. Those drugs deplete all of your B cells, whereas what we're thinking about doing is a much more precision-based approach. The third thing we're excited about is that we can do this much faster than traditional drug discovery. And that's not because we're so smart, it's because the search space is 2 orders of magnitude smaller. When we think about targeting a protein, you have to pick the protein. There are 20,000 proteins in every one of your cells, minimum. In contrast, there are only a few dozen cell types in any given tissue. And so we can canvas the search space much more effectively in a much shorter amount of time and find what are the real causal pathogenic cell populations. The last reason we like this approach is that unlike a standard protein blocker, we don't have to dose chronically. If you're on any sort of medication, you probably know you have to take a daily pill or a weekly injection, and you have to deal with any side effects that come with that chronically. In contrast, it takes us a week or two to get rid of these cells. It's 1 or 2 doses. You'll see in the deck, we've been dosing all of our animal models with 2 doses, 2 weeks, and we're done. We have to dose again when the cells come back. Now, the rate of cell replacement is going to vary by disease, but we're talking about months, if not years, because the diseases that we're targeting, unlike cancer, develop over years or decades. You can almost think about knocking back the biological clock of disease progression by a pretty substantial degree. So that's both good from patient experience. You don't have to take a pill every day, but it's also good for therapeutic index because you won't have to deal with the side effects of the drug being on board or in your body for most of the time. The drug won't be there.
Yeah, Adam, on the faster discovery, is it reasonable to think that you can also achieve faster discovery because of how AI tech, et cetera, has advanced?
Most definitely. I mean, the way that we select targets is very much machine learning driven, and we've employed a lot of those strategies, big data, and all of the above in order to accelerate that process.
And when you said earlier, there's 20,000, see if I get this right, proteins in your cell, it's the pathways to that protein that make it daunting, right? That there's
No way. Yeah. So the proteins each protein does something. They're like little tiny molecular machines inside your cell. They bind to each other. They trigger these sequence of events that we call signaling pathways. And ultimately, they end up with the cell doing something different. And so when we block proteins, we block the activity of whatever that molecular machine is doing, we're trying to throw a wrench in the gears of the overall system. And if this were a human design factory, you could probably do this pretty effectively because each component of this thing would be non-redundant. It would be doing one specific job and nothing else would do it. The problem is cells are not human designed. They are wildly redundant systems. And so when you block one protein, there's usually 5 or 6 others that are ready to jump in and take its place.
Yeah. And by contrast, when you're targeting the cell specifically, it just decreases your, forgive me for the pedestrian views of these, I'm just trying to, but it reduces your audience and you can target it better.
Exactly. So when we are whenever we block these molecular machines, we're trying to change cell behavior because cells really are the functional units of your body, of each tissue. You're made up of cells. And so whenever we're blocking these proteins, we're trying to get cells to do different things, or usually to stop doing what they're doing if they're a pathogenic or disease-causing population. But often it's not sufficient. So what we do at Arda is we just say, well, if these are bad cells, if they shouldn't be there, then let's just get rid of them. This is not applicable to every single disease. There are some diseases that are caused by general cell death, and their depleting cells isn't going to help you. But there are a lot of diseases where the disease is driven forward by the presence of a population of cells that's doing something it shouldn't be doing. And that's exactly.
Yeah. So since you brought it up, so we have a question about, does this technology work with MS patients?
So MS is a great example of a B cell depletion strategy. So MS, as far as we understand it, there aren't that many good therapies except for these drugs that deplete B cells like rituximab. Rituximab is actually pretty effective in MS. Not completely. There are definitely subpopulations of relapsed remitting MS that don't respond. But that is exactly the kind of situation where if you can get rid of the B cells for many patients, that will ameliorate the disease. Now, MS isn't really on our radar because this drug already exists and seems to do a pretty good job.
Makes
but
sense.
There are a lot of other diseases where that would be a similar conceptual strategy.
Yeah, great. Perfect.
Okay, so if you're new to the cell depletion space, you might not know just how broadly applicable this strategy can be. And so we've spent years compiling what I think might be the largest library of cell depletion literature that exists outside of cancer. And here's just a few examples. But the point of this slide is to demonstrate that this is not a single disease or a single target approach. We have evidence that this works across diseases, across tissues, cell types, targets, and modalities. Here we just have a few examples, right? So atherosclerosis, pulmonary fibrosis, asthma, cardiac fibrosis, using antibodies, CAR Ts, small molecules, or suicide genes. Really, we think, and the literature supports, that this is a broad opportunity. The thing that enables this to happen now, whereas previously it was just, I think, an interesting idea, is that we finally have enough single cell data to do targeted cell population discovery and depletion correctly. So if you think about it, if you're trying to target a subpopulation of fibroblasts in order to treat fibrosis, can't get rid of all the fibroblasts. Unlike B cell depletion, getting rid of all your fibroblasts would be acutely toxic. The same would be true of T cells, endothelial cells, epithelial cells. So we have to find subpopulations. But we did not have the technology to identify subpopulations of cells with high resolution until maybe 10 years ago, with the advent of technology that we call single cell sequencing technology. Now, we didn't invent single cell sequencing technology, but we certainly are benefiting from it. It took 10 years for this technology to reach maturity. So the first data sets were from a couple patients here and there, a few thousand cells per patient. Now we have data sets of hundreds of patients, millions of cells, and that allows our ML algorithms to do the work, right? And to do it the right way.
So why were they anxious to discover this identification of the cells prior, independent of what you're doing with them?
Yeah, what motivated the development of the technology?
Yes. Thank you.
I think everyone has realized in the biology field for a long time that counting up cells the way we historically have been able to count up genes would be an incredible benefit to understanding general disease biology. And if you're interested in understanding this technology further and what people use it for outside of what Arda uses it for, just go into Google and type, single cell RNA sequencing and your favorite disease. And what you'll find is that people use this technology to say, there are cell types in this disease that we never knew about. These cells interact with these other cells in ways we didn't understand. This cell type A changes into cell type B. It's essentially delivering an understanding of the layer of cell biology in diseases that we previously didn't have. We previously really only had almost a gene level understanding of these different diseases based on pre-existing sequencing technology.
Yeah, totally makes sense.
So
Great.
then, having hopefully motivated a little bit of the core concept, let me jump to a quick description of the team. So I'm Adam, as I mentioned, a scientist by training and CEO of Arda. My co-founder and chief technology officer, Remy Leberge, has been in life sciences for about 20 years. We first met when I was a PhD student and he was a postdoc in the same lab. And that's when we first started talking about this idea. In fact, Remy went on to become one of the first employees at a company called Unity Biotechnologies, which was developing small molecules to deplete a certain kind of pathogenic cell. He was there for about 8 years and helped bring 2 different drugs to the clinic. And the successes and the failures in that space have really helped inform us and think about how we can do it in a more broad way at Arda. Our third member of the leadership team, Scott Turner, joined us only a month ago. He's currently our chief scientific officer. Previously, he was the chief scientific officer at a company called Freund of Therapeutics, which is developing drugs for pulmonary fibrosis, which happens to be our lead disease area as well. So his experience incredibly relevant. He helped bring multiple programs to the clinic, including their small molecule drug for IPF, which has now achieved a successful phase 2A and is enrolling in a phase 2/3 trial right now. So plenty of relevant experience on the leadership team. We also have a great set of individual contributors as well on the team, an advisory and consulting board that is excellent in terms of both disease biology as well as antibody and antibody drug conjugate development expertise. And then on the investing side, as Becky mentioned at the beginning of the call, Andreessen Horowitz led our seed round. We also had participation from GV, Village Global, the Longevity Fund, and others. And on the Series A, Andreessen will be leading that. We also have participation from GV, a little bit of new money coming in with much more expected to be committed, hopefully in the next few weeks. And ideally, some members of your group.
Yeah, terrific. So continue. I'm sorry, I have a question, but it can wait. Go ahead.
Oh, I'm happy anytime.
Yeah, it's not having to do the team with it's more macro in terms of what, presuming you succeed at this objective, how dramatically does it change the incumbent way of fibrosis, for example, is being treated? Like, how big of a sea change will there be in time, money, treatment, success? Do you have a sense of that?
We do. I mean, we've certainly done market analysis on our lead indication areas, which are lung fibrosis, liver fibrosis, and an autoimmune disorder. And each of these are multi-billion-dollar markets per year, right? And so just from the initial disease areas alone, we're looking at very large markets. But I think perhaps more broadly, which may be what you're getting at, is will this really transform the way we think about these diseases? And of course, there's plenty of ways drug development can go wrong. But if this approach, as Becky said, big if true, if this really does pan out the way that we think it will completely change the way that we think about treating these diseases, right? They will no longer be a pure, let's get you on a drug to hopefully slow the progression and manage the disease, but this disease is going to be relentless going forward no matter what we do. Here, we really have the opportunity to stop these diseases in their tracks. Now, in not all cases will we be able to fully cure them, right? It's like we can't necessarily turn the clock all the way back to the original thing that caused the disease, which can sometimes precede the development of symptoms by decades. It'd be great if we could, but that's not really on the table for things like pulmonary fibrosis. But what we can do is when these patients come into the clinic, if it works, we'll have something to give them that can truly give them back their lives. They go in, they take the drug for a couple weeks, they go back about their lives, and there's nothing they have to do. Maybe 6 months later, maybe a year later, they come back in, they dose again. But what we're hoping to achieve here is drugs that are not just incremental, that are not just a little patch on top of whatever's already happening, things that are truly transformative.
And bringing back quality of life type metrics, right? To meaningful way.
Absolutely.
So it sounds like, yeah. Yeah. So thank you for articulating that.
And so to tell you just a little bit about what we actually do, we think of going from a disease of interest to a validated molecule as quickly as we possibly can. Because we're trying to capture a large amount of opportunity space here, we think there's many indications where we can employ the strategy, and we want to bring as many of them into our pipeline as we can, given our current bandwidth. And so we've developed a system, which we call the Arda product engine, that goes from target nomination, which is to say, how do we actually tackle this disease, all the way through to program development in as short a time as possible. And so we start at the top of this circle. Target nomination for us really means first, we have to find the right cells to eliminate, pathogenic cell identification. Then we have to find the surface markers on those cells that allow us to home in on them, sort of like a tag on the surface of these cells. We can find usually a small number of those that pass our filtering criteria. We generate assets or antibodies, these are actual molecules, that allow us to bind those vectors. Those molecules we engineer to have cell-killing activity. We test them in a in vitro or cell culture model system to make sure they work as expected, and then we move them into our preclinical in vivo or animal models. And there we're looking for three key criteria to advance the program further. Can we get rid of the target cells, shown here? Can we avoid depleting other cells? So it's like, is it sensitive and is it specific? And the third thing is, can we improve disease endpoints? Does this actually matter? And if we hit all three of these checkboxes, we move forward into program development. If we do not, then we stop. We move on to the next thing. And we've run this circle multiple times, and we've learned quite a lot about the rules of this process in the non-oncology, the non-cancer space. And it turns out there's a lot of proprietary know-how to be gained here. And frankly, we've learned a lot of it the hard way. But increasingly, we have a good sense of what makes a good target, what makes a good modality, how do we move this forward quickly, how do we gain sensitivity and specificity. And I think we're well poised to continue to expand the pipeline.
And is this something that you would do simultaneously in multiple target nominations, or is it so overwhelming you have to do one at a time?
So we think about doing this on a per disease basis. And so each disease is a different target. Many times we'll start with half a dozen targets for that disease, but then we'll narrow down into the most effective one. Because in general, we don't want to take multiple drugs forward for the same disease, because that ends up being a little bit internally competitive.
And then doing multiple disease at the same time with multiple targets is just a bandwidth issue, money issue, both?
Primarily a money and bandwidth issue. Yeah, those sort of end up being the same thing at some level. And I think that it's very hard for a small biotech company to prosecute more than a handful of programs at the same time. We have a slight benefit in that our approach is quite a bit simpler in some ways than blocking a protein and trying to figure out how the cell changes its behavior. Whenever you're trying to change cell behavior, there's almost a black box of biology understanding that we need in terms of what does that protein interact with? What does it do? Where does it go in the cell? There's all of this stuff we have to figure out. At Arda, we don't care what the proteins do. They're just tags. We bind them and we kill cells that express them, and that is it. And so because of this, when we select targets, getting to an antibody that binds that target and kills the cell is a very quick process, on the order of a few weeks, few months. Right? This is not a multi-year process, which it is for the standard approach in most cases.
Would you, and I don't want to interrupt your flow, and I want to make sure you get to everything, but it struck me that being that you're doing this in what arguably is a novel way, and this being attacking the cell, is the FDA, they're smart people, they're sophisticated. Is it going to be a learning curve for them to approve this sort of thing? Or are they on board and understand what you're up to here?
It's a great question. So one of the reasons why we're focused on using existing modalities like antibody drug conjugates is that the FDA has tons of experience with these. Those are
Good.
in the cancer space, right, for the most part. But thinking about what kind of efficacy do we need to see? How do we think about the safety profile of these? That's something with lots of precedent. And so we're pushing the envelope slightly by going into these new indications, but it's also one of the reasons why we are selecting indications where the morbidity profile is as bad as many cancers. So I'll show this in a slide or two, but pulmonary fibrosis, if you had a choice between non-Hodgkin's lymphoma or pulmonary fibrosis, I'd pick non-Hodgkin's lymphoma every time, right? Because pulmonary fibrosis is a terrible disease and has like a 3.5-year median survival to diagnosis, worse than many cancers.
Wow. That in layman's terms, that's lung cancer.
Pulmonary fibrosis is
Yeah.
actually not cancer. It's a type of it's a type of fibrosis, which is a general term for diseases that cause the stiffening and mechanical failure of your tissues. The one that people are most familiar with is cystic fibrosis, which is the one that's caused by a single gene mutation. And that one, the right approach is probably a genetic modification tool, right? Because it is caused by a single gene mutation. But pulmonary fibrosis is also lung fibrosis. Cystic fibrosis is a type of lung fibrosis. But instead of being caused by a genetic mutation, it's caused by a combination of factors that we don't really understand. What happens is we think a lot of it's environmental. There might be some genetic component to it. But in your 60s or 70s, generally men, will start to experience shortness of breath. And that shortness of breath will progress until these patients are on oxygen. They can't go about their day. And ultimately, they'll require lung transplant in order to survive. And that is caused by what is essentially a chronic wound healing response happening in your lungs. So you've ever gotten a cut on your arm, and especially if it's a big one, you've seen the scar forming process occur. Imagine that process going on for years without stop inside your lungs. So at the end of this process, you have all of this literal scar tissue that's preventing your lungs from doing what they should do.
Yeah, that was a very good explanation because I understood it. Thank you for that. I get it. Thanks.
And so we think, as I've said a couple times now, that this approach has broad therapeutic potential. We're starting with pulmonary fibrosis, and I have a slide in just 2 slides that sort of goes into this disease in a little bit more detail. But every disease on this slide, we have looked into human single cell data, and we have seen evidence of a pathogenic population. And this is another thing that I think differentiates Arda's approach. When we select targets, the data that we use always starts from human data. And what that means is we take tissues, we or others, if we're utilizing their data, take tissues out of patients, and usually within several hours, right, as quickly as we possibly can, get that into a machine that sequences the cells and gives us data back. So this is as close as we can get to something that is representative of what's going on inside of patients' bodies. Our pipeline as it currently stands is 2 programs in pulmonary fibrosis, one of which is our lead, ARDA1, and one of which is our backup, ARDA2. We have a program in liver fibrosis, which has reached some really exciting in vivo data that I'll show you in a few slides. And then we have an autoimmune program where we selected targets and are able to repurpose some of our existing molecules because it turns out the targets overlap. This is another benefit of what we're doing. We find that in many cases, the same targets show up again and again. And that's because these cell states, even when we're in different diseases, they themselves are not that different because cellular biology can be so conserved in different disease contexts. And then we have a number of other disease areas that we're excited to explore, which we'll call ARDA 5+. We're mostly pursuing in-house drug development, but we also have a lot of opportunities for partnering. And we're in active conversations with a number of large pharma companies about this. So we look at a couple different possibilities in this space. We could do co-development deals of existing assets. We could do discovery deals where we help pharma companies find their own novel targets for new diseases. And we've seen some interest on both of these. I think people are increasingly excited about the idea of targeted cell depletion because there has been recent success in the B cell depletion space. It's causing a lot of pharmas to ask, well, where else can we apply this? And on the co-development side of things, we think our focus on antibody drug conjugates in particular is a modality that pharmas are very excited about right now. If you followed the news in this space at all, we've had a number of antibody drug conjugate approvals over the last 5 years. because we've gotten a lot better as a field at making these safe and effective. And so we're able to ride some of the headwinds of that. And then lastly, we have licensing opportunities, although that will probably occur once our programs are a little further along, likely in the clinic.
So this slide also applies to, from an investor perspective, potential not only growth and revenue opportunities, but one would immediately think that there's an exit to a big pharma if you're wildly successful, right?
Absolutely. And that's one of the reasons why partnering is a big part of our strategy long term.
Yeah, great.
Okay. So jumping into our lead indication, pulmonary fibrosis, as I mentioned, it's the thickening and scarring of lung tissue. It leads to a host of symptoms, ultimately culminating in lung failure. We chose pulmonary fibrosis as our initial disease area for a few reasons, all of which are because it's a great fit for our approach. The first is we already had a pretty clear idea of the cells we wanted to target, just because we have a strong background in this disease. I've worked on this disease for years. My co-founder, also our CSO, all of us have a lot of experience in this disease area. So we generally knew where we wanted to go, and we expected the data to lead us in that direction, and indeed it did. There was a lot of high quality data already existing that we could work with by employing our novel algorithms and our novel computational methods. This disease has significant unmet need, which I already mentioned, 3.5-year median survival to diagnosis. However, it also has a clearly defined clinical development path because there are 2 approved drugs. Those 2 drugs each do $2 billion in sales a year, despite not working very well. Right? If you actually look at the survival data since those drugs have been approved, and it's been probably about 10 years now, they've barely shifted the needle in terms of overall survival of this disease. They're also very poorly tolerated and have to be taken 2 or 3 times a day. So lots of opportunity for better drugs.
So when you say it hasn't improved, it might have been, 3.2 years, now it's 3.3, and I'm not backing you in, but that's what you mean, right?
That's exactly what I mean.
Yeah.
So our strategy for this disease is to get rid of the fibroblasts that produce collagen and ECM. So just to give you a very high level overview of how fibrosis works, and this is true irrespective of the tissue, but in this case, we're talking about lung. It starts with injury, and this can precede the development of symptoms by years, if not decades. That injury leads to a feed-forward cycle of inflammation and activation of these fibroblasts that looks a lot like a wound healing response, but it never shuts down the way a wound healing response should. What those fibroblasts do is they contract the tissue and they deposit collagen that stiffens the tissue. It is very analogous to watching your wound close up on your skin as it heals.
Yeah.
Over time, this causes tissue stiffening, functional decline, and ultimately death. We are not the only company that has thought about blocking the activity of these cells as being the key step in this process that will allow patients who have actively developed symptoms to stop further progression, right? Lots of people have realized this. The difference is they're trying to develop drugs that block individual pathways in order to tune down the activity of these cells. This is marginally effective. The 2 approved drugs do this, right? So it actually, in many ways, supports the clinical hypothesis, but this is a very, very stable cell state. And it's a very stable cell state because it thinks it's doing something good. I mean, not to anthropomorphize the cell, but this is a process that in other contexts would be beneficial. And so there's a lot of biological pressure going into keeping this process going. But in this case, it's not beneficial. It's absolutely maladaptive and is causing the disease to go forward. And so in this case, rather than blocking an individual pathway or protein, getting rid of the whole population is likely, just from first principles, to be much more effective. And indeed, that's what we've seen borne out in the data.
And wouldn't it also be true that the other therapeutics attacked, in your example, 3, but there's probably 30, right?
That's exactly right. So each therapeutic only hits 1, and there are dozens of pathways that contribute to this cell state. So not to go too technical, but just to give you a sense of the kind of computational analysis that we do at Arda, we start with single cell data from human patients. And this is a certain kind of plot where it's made up of a series of tiny dots. There are hundreds of thousands of dots. Every dot represents a cell that was previously in the lung of a patient. We sequence those cells to figure out what they're expressing at the genetic level. And then we place them in this plot in a way that their position is informative of how similar they are to other cells. So cells that are close to each other are similar. Cells that are far apart are less similar. And so you can immediately see that they separate into these big clumps, which we call clusters, and which are colored by those different major clusters. These represent major cell types. For example, B cells, T cells, fibroblasts, endothelial cells, things like that. We hone in on just the fibroblasts, which is this little cluster right here. And we ask two things. Do we find evidence of a subpopulation of fibroblasts that is enriched in disease, which means it's not present in healthy people, it's only present in patients? And do we see that those cells express factors that we know drive the disease forward? And if we see both of those things being true in the same cell population, then we have a strong hypothesis that that population is detrimental. And so the disease enrichment analysis is shown here. There's actually a lot of proprietary computational work that went into this particular process, which I'll sort of gloss over for the time being. But suffice to say, it's very different than the way most people analyze this kind of data. And then here's the pathogenic gene expression analysis, where we are looking for the expression of fibrillar collagens and extracellular matrix proteins, which are the exact proteins that cause the tissue stiffening. So again, very simple, right? We're not trying to be too fancy with our disease hypothesis here. We just want to get rid of the cells that produce the actual proteins that cause the tissue to stiffen. And so we find that population, and then we go looking for surface markers that allow us to kill that population without killing other cells. And so here we've identified a target, which we'll call target 1, because we're under non-confidentiality in this case. And just to walk you through this, this is another one of these single cell plots where every dot is a cell. Here's that fibroblast population. You can see these in blue are cells from patients who are healthy, control patients. You can see that there is this little blob up here that is not present in controls, but is present in IPF patients. It sort of looks like Virginia, maybe? Anyway, we can call it Virginia. So in this Virginia blob, we have cells that are almost entirely from IPF patients and not from controls. So this means they're enriched in disease. Then we also ask, what do they express? Do they express things that are bad? That's this pathogenicity score. And you can see again that Virginia has pretty much the highest level of this pathogenicity score. And that allows us to annotate these cells as our target population. We then find target 1, which is an actual protein that's on the surface of these cells, as being reasonably sensitive and specific for this population. Now, it's not perfect, and no target ever is, but it doesn't need to be perfect. It just needs to be sensitive and specific enough. Yeah, go ahead.
So you're going to get rid of some of the good ones, but most of the bad ones. And so it's exponentially better than how it's done now.
Exactly. Exactly. And I think one thing to point out is understanding where the best trade-off between sensitivity and specificity lies, exactly as you just said. How many bad ones do we have to get rid of? How many good ones can we afford to get rid of? That helps us decide what the best targets are. Yeah. And this is something that we had to learn internally, because what makes a good target in this kind of situation is very different than what makes a good target in the cancer setting.
Yeah.
And so again, I know this gets a little bit technical, but one of the questions we get a lot is, okay, well, you've shown us that this protein is, or this target is expressed at the RNA level, but antibodies bind proteins. So is this thing actually expressed at the protein level? And what this slide is meant to represent is just, yes, indeed, that is what we see. So by measuring by flow cytometry, which is one measurement of the cell surface of a cell, we can see expression of this protein. This is immunohistochemistry, which is another way of looking at it. Again, in brown, we see the expression of this protein in the lungs of fibrotic patients. We can see that this target is upregulated by TGF-beta signaling, which is a core pathway involved in the disease. So this is all consistent. And then lastly, we see that the target, which the expression of the target is shown here on the x-axis, correlates quite well with the expression of fibrillar collagens shown on the y-axis, which is a measurement of disease activity. And so in this case, every dot is an individual patient. And what we're seeing is that the patients who have more disease activity express more of the target. And that means they are more likely to benefit from our drug.
So sufficiently close enough.
Sufficiently close enough, exactly. And so we developed an antibody drug conjugate against this target. Now, this is a type of therapeutic that deserves a little bit of explaining. So what antibody drug conjugates are is they are an antibody which binds proteins that is conjugated or linked to a cytotoxic payload. It's a molecule that kills cells, sort of like a chemotherapy. But unlike a chemotherapy, which goes throughout your entire body and kills any cell that it happens to go into, this modality causes that molecule, that cytotoxic agent, to only go inside cells that express the protein the antibody binds to. So here's your antibody. It comes in, it binds to its target, which is this protein on the surface of the cells. That triggers internalization. So the whole thing gets brought inside the cell and is then degraded. Once it's degraded, that cytotoxic agent, that chemotherapy, is released and kills the cell. So people have described this, a concept which has been around in the literature for decades, as the magic bullet of chemotherapy, because it does exactly what you want chemotherapy to do without the systemic side effects. Now, in practice, it doesn't work perfectly, but it does do a lot better than actual chemotherapy. The first ADC was approved in 2000. 13 of them are now approved today, 7 in the last 5 years. So there's been an accelerating trend. And the reason for that is that in the last few years, we have gotten much better at making these things safe and effective. And there's just a few improvements that I've listed here on the bottom right of the slide that explain how we've gotten better. So all parts of this molecule have been engineered. And indeed, at Arda, we are incorporating every single one of these state-of-the-art technologies in order to make the most effective, safest antibody-drug conjugate we can.
And the ones that have been used, while haven't been perfect, they've been better than traditional methods of just going in and getting chemotherapy.
That's exactly right. All of the ADCs that are currently on the market are what we call first-gen ADCs, and they're already better than chemotherapy, but they are inferior to what we're generating now in clinical development. And the next round of ADCs that get approved are going to be even better. There's been a couple, 2 or 3 in the last few years that are sort of maybe second-gen ADCs, but we're really in the 3rd-gen, possibly even 4th-gen stage at this point. So we made an antibody-drug conjugate against this target. We'll call this antibody-drug conjugate ARDA1. It hits target 1, so easy to remember. And these plots just show that it binds its target, and it binds both the mouse and human version of its target, which is important. It internalizes. Not every target does, but this target happens to internalize, which is good because we need it to go inside the cell to deliver the chemotherapy. And it actually is cytotoxic. It kills the cell. So here you're looking in red at cells that express the target, as compared to in black cells that don't express the target. And so you can see that the killing really depends on the expression of the target.
Yeah.
We tested this in a mouse model of pulmonary fibrosis. So with all drug development, you have to start with animal models. In this case, it's not a perfect representation of the human disease, but it's actually pretty good. The way this works is we injure the lungs of the animal with this molecule called bleomycin. And over about 21 days, so much faster than the human disease, which is necessary for development, it turns into, from a normal lung, shown here, which is mostly airways and open space, to this densely fibrotic lung, where collagen is shown in brown. And that's what we're trying to prevent as much as we possibly can. This is, I should point out, a very aggressive model, very difficult to have any effect in this model at all. So we identified this target, target 1, and we developed ARDA1, as I mentioned. And here we're showing that it works in this model. So target expression is just whether or not we can actually deplete the cells. In black is your baseline. These are healthy mice that don't have any lung injury. In purple are mice that have, their lungs have been injured, and they've been dosed with the control ADC. So you can see the expression of the target goes way up because these cells are being created. When we dose with our ADC, ARDA1, we're able to reduce this significantly, not quite back down to baseline, but pretty close. This, at the same time, reduces histological fibrosis, which is the relevant disease endpoint. So this shows we can actually prevent the fibrosis. It reduces lung weight relative to body weight, which is a good sign because generally these lungs increase in weight by quite a lot because there's all of these cells that are sort of flowing into them, inflammatory cells, fibroblasts, things like that. We reduce collagen mRNA, which is a measurement of fibrotic deposition, and we reduce Ashcroft score, which is a standard metric for fibrosis in this disease.
So in the first target expression, again, very, myopic, I say it that way because I don't have a lot of information prior to this. It brings it almost back to normal.
Use that one more time.
No, in the target expression where you use ARDA1, it brings it almost back to normal.
Yeah. Exactly. Yeah, it is. And you can actually see here, just visually, kind of what this does. So this is a healthy lung. These things here are airways. And so they have a little bit of collagen around them, which is good. That's good collagen. But what you're seeing is when these mice develop disease, the entire tissue becomes more dense and it develops collagen outside of the airways, shown in red here and in brown here. You can see how much brown there is relative to here. When we dose with ARDA1, we reduce that quite a bit. Now, it is not back to perfect, right? We are not fully curing this disease, but we're having a major effect. The collagen surrounding the airways is still there, so we haven't gotten rid of the good collagen, but we've gotten rid of a lot of the bad collagen. Okay, so I'll now move on to our next program, our next indication area, which is liver fibrosis. Liver fibrosis most often occurs in conjunction with a disease called metabolic dysfunction-associated steatohepatitis, which is a mouthful, so we'll just call it MASH. MASH is essentially fibrosis that arises due to excessive obesity and the liver damage that arises because of it. It's a huge market. There are a lot of people who have this disorder in the United States, and fortunately for that patient population, we have recently seen some successes. So Ozempic, which is a GLP-1 agonist, Wegovy, another GLP-1 agonist, same drug, actually, just dosed differently. Those have a big effect on weight loss. And that, over time, will help a lot of these patients. However, advanced patients will not benefit from these diseases. In fact, when you get to the end stages of liver failure due to liver fibrosis, you start to sort of lose weight without meaning to. And that's a very bad sign. In those cases, taking a drug that causes you to lose more weight would be detrimental. What we want for those patients is to prevent the fibrosis, possibly even reverse it, without causing a lot of metabolic changes. In addition, the GLP-1 agonists, they don't seem to reverse fibrosis at all, even in earlier stage patients. And so that's another opportunity for indication expansion.
Yeah.
So our strategy here is not too dissimilar from what we did in the lung, which is the liver becomes full of excess lipids or fat. That causes the hepatocytes, which are the primary cell type in the liver, to become damaged. They release a bunch of signals that causes immune cells and these cells, which are called hepatic stellate cells, they're like the fibroblasts of your liver, to activate and start trying to fix the wound. It's a very similar concept to the lung that we talked about earlier. Unfortunately, they can't fix the wound because the liver is still fatty. And so they end up depositing too much collagen, and this actually accelerates the problem. And so now you have cirrhosis, liver failure, even hepatocellular carcinoma, which is cancer. And so our strategy was to deplete these cells and see if that made a difference. We identified a target 3, which is a good marker of these activated hepatic stellate cells, and we tested it in a mouse model of diet-induced obesity. So if you're going to be a lab mouse, this is not a terrible lab mouse to be. You get fed a delicious high-fat diet for 10 months, and you put on all kinds of weight, and you don't notice it, but your liver progresses towards failure because of how much weight you're putting on. And so we get activation of these cells, and then we dose for just 2 weeks. There's a very short dosing window relative to how long it takes the disease to develop, just to see what can we do in a short dosing window, because often these late-stage patients, they don't have very much time. And so we're able to reduce the target. This reduces collagen expression, which is what we expect because we're reducing, that is what these cells produce. And we almost normalize alanine transaminase. Now, you may not be familiar with alanine transaminase, but if you've ever gone to the doctor and had a liver function test, this is one of the 2 enzymes they measure. So it's exactly the same enzyme in humans as it is in mice. It goes up with injury, so higher is worse. Our ability to bring this back down to almost normal level in 2 weeks is, I believe, unprecedented. I don't think anyone has seen any effect this fast or this big. You can reduce ALT by losing weight over time, but it takes months. And the ability to do this in 2 weeks without any weight loss, and these animals exhibit no weight loss, is a complementary approach that could be very beneficial for patients for whom the weight loss is not an option.
And that's sustainable without ongoing doses, or
so we don't know
less
how
periodically, right?
Right. So we have not yet measured how long it takes these cells to come back.
Yeah.
But given that it takes 10 months for them to develop in the first place, we think there's a pretty good chance it will be quite a long time.
Yeah.
At the same time, we increase albumin, which is a marker of hepatocytes. So this is a sign that we may actually be causing the liver to regenerate. So maybe we're not just slowing disease progression, maybe we're actually reversing some aspects of it.
Yeah.
And so getting towards the end here, here are our program timelines over the next 2.5 years. We're raising our Series A right now. As I said, we hope you'll be a part of it. That money will take us through the end of 2026, at which point we'll raise our Series B. In that time, we will reach development candidate selection, which is what this little diamond is, DC selection, for 3 programs. What is DC selection? It is saying we have finalized the design of the molecule that we're going to take forward. It's a very important preclinical milestone, and it adds substantial value to a company to have DCs, especially multiple. For our lead program, in addition to DC selection, we'll actually get all the way to IND filing, which is when the FDA says you can put this drug into patients. And so that is another very important milestone. And if things go well, we might even get to our first in human doses. There's still a long way to go before these drugs are approved. However, in general, these are major milestones, and they unlock additional partnership opportunities, additional fundraising. The way we see this going long-term is we'll raise the Series B to fund the clinical trials. Clinical trials are very expensive, and so this will likely be tens of millions, probably close to $100 million Series B. But with those funds, we will likely achieve clinical data that suggests efficacy, and that may open the road to an exit, either an IPO or an acquisition. So you can think of the Series A as potentially the second to last financing round. Obviously, we can't make any formal promises there, but that's kind of how we're thinking about it.
Yep. That's super fast.
And so in summary, we'll take this Series A funds, we'll use it to progress 3 programs to DC selection, 1 reaching IND. We'll expand into 2 to 3 additional programs. We'll further produce and expand the platform, and we'll go from there. And that is the end of my presentation. Thank you.
Yeah, that was super, super helpful. And for not that it matters, but I really enjoyed learning about that. So let's just repeat the ask one more time, and then we'll wrap it up and do our follow-up.
Absolutely. So we're raising a $40 million Series A on a $40 million pre-money, so 80 million post. We have 25 million of the 40 million committed, and we're looking to raise an additional 15.
Great. Thank you, Adam. That was really, really informative, and I appreciate having you here, and also for Shelley and Mercury giving us the opportunity to share this with the community, and we're going to make sure we share it with others. So thank you again, and we'll make sure everybody who participated in those that RSVP'd and others get in contact with you directly, and then talk about posting the recording and so forth. So thanks again for doing that. Really appreciate it.
Thanks everyone for your time and attention.
And thank you everybody for joining us. And as I always say, thank you for sharing with us the only thing you can't make more of, and that's your time. Till next time. Thanks. Thanks, Adam.